Nearly every trait we possess, from hair and eye color to height and shoe size, is determined by our genes. Genes are small segments of DNA that code for proteins. Proteins perform many functions in our body. Some proteins serve as components of our cell membranes. Other proteins make up the structures of our bodies, like muscle, skin, and fingernails. Still more act as enzymes, catalyzing reactions around our body. When you break them down, proteins are made up of amino acids placed in a very specific order by cellular organelles called ribosomes. The specific order of amino acids determines the folding pattern of the protein and therefore the very specific function that the protein will perform. Our DNA is essentially a set of blueprints for building the proteins that compose our body. Within the genetic code of the DNA are the instructions for the correct amino acid sequences to build proper proteins. But how do our cells convert the messages stored in DNA to the proteins our body needs? Two problems stand in the way of accomplishing this task. First, the DNA is stored in the nucleus of the cell, while the ribosomes responsible for building the proteins are found in the cytoplasm. How do the DNA instructions get from the nucleus to the cytoplasm so the ribosomes can read the instructions necessary to build the proteins? The second problem lies in the fact that DNA speaks the language of nucleic acids, while proteins speak the language of amino acids. How can the language of DNA be converted to a language that ribosomes can understand? The solution to both of these problems lies in a single molecule, ribonucleic acid, or RNA. RNA is a nucleic acid just like DNA, but there are a few key differences between these two molecules. First, while DNA is a double helix, RNA is only single-stranded, so it forms a single helix. Secondly, as the name suggests, ribonucleic acid, or RNA, contains ribose sugar in its structure, while deoxyribonucleic acid, or DNA, contains deoxyribose sugar. The difference between these two sugars is the presence of an extra oxygen on the ribose sugar. Another difference between DNA and RNA lies in the nitrogenous bases they possess. DNA contains adenine, guanine, cytosine, and thymine, as you already know. RNA, on the other hand, contains adenine, guanine, cytosine, and uracil. Uracil is another nitrogenous base that takes the place of thymine in RNA. Uracil pairs with adenine in RNA, just like thymine does in DNA. Finally, one last difference is that RNA is much shorter than DNA. While DNA contains thousands of genes, RNA contains the information for only one gene. RNA is a special molecule able to solve the problems associated with getting from gene to protein. While DNA is stuck inside the nucleus, RNA is able to travel from the nucleus to the cytoplasm to bring the genetic information to the ribosomes. RNA is also bilingual, speaking the language of DNA and of proteins. RNA is therefore able to translate the DNA instructions into an amino acid sequence to build the proteins of your body. There are 3 different types of RNA that each play a role in the process of taking genes to proteins: messenger RNA, or mRNA, transfer RNA, or tRNA, and ribosomal RNA, or rRNA. Let's take an in-depth look at the process of protein synthesis and examine the role of each type of RNA. In order to make a protein, the first thing that must be done is to convert the genetic message in the DNA to a mobile form that can move from the nucleus into the cytoplasm to find a ribosome. This process is called transcription. Transcription occurs inside the nucleus and involves copying the information from a single gene on the DNA into a molecule of messenger RNA, or mRNA. During transcription, an enzyme called RNA polymerase binds to a promoter site on the DNA. The promoter site acts as the start signal for transcription and marks the beginning of the gene of interest. Once RNA polymerase binds to the promoter site, the enzyme unwinds the DNA helix to expose the nucleotides of the gene. RNA polymerase then reads the DNA strand and adds complementary nucleotides to build a molecule of messenger RNA. RNA polymerase moves down the DNA strand and the messenger RNA grows. Behind the polymerase, the two DNA strands close, reforming the double helix. Once the polymerase reaches a stop signal at the end of the gene, the messenger RNA is released and transcription is complete. Let's take a closer look at the base pairing that occurs as a strand of messenger RNA is formed. Remember that in RNA, uracil pairs with adenine instead of thymine. Looking at the DNA strand, the first base is cytosine, represented by C. C pairs with G in RNA just as it does in DNA. The next letter is A for adenine. Remember that in RNA, adenine does not pair with thymine. Uracil replaces thymine in RNA, so in this case, A will pair with U. Just like in DNA, G pairs with C. T will pair with A. Again, we have an A that would normally pair with a T, but in RNA, there is no T, so we use uracil, U, instead of thymine. G pairs with C, G pairs with C, just like in DNA. Once the messenger RNA has been created during transcription, it must move from the nucleus into the cytoplasm to find a ribosome. Remember that ribosomes are tiny cellular organelles responsible for manufacturing proteins. Ribosomes are made up of ribosomal RNA, or rRNA for short, and protein. There are thousands of ribosomes found in the cytoplasm of every cell. Their job is to read the messenger RNA and help translate the genetic message into a protein. The process of making a protein from messenger RNA is called translation. Translation occurs in the cytoplasm, where a ribosome binds to the messenger RNA. The ribosome reads the messenger RNA 3 nucleotides at a time. A set of 3 nucleotide bases on a strand of messenger RNA is known as a codon. Each codon translates into one amino acid in the protein. A codon is like one word in the language of nucleic acids, while an amino acid is like one word in the language of proteins. This process is called translation because we are literally translating one language into another. Just like English can be translated into Spanish and vice versa, the language of nucleic acids, like RNA, can be translated into the language of proteins. The key to translating one language to the other lies in the universal genetic code. This chart summarizes the universal genetic code. It may look complex, but once you know how to read it, the chart becomes a simple and useful tool. In the center of the chart are the names of the amino acids coded for by each codon. In this case, the names are abbreviated. The 3 bases of the codons are found on the margins of the chart. The name of the correct amino acid is found where the 3 letters of the codon intersect on the chart. The first base in a codon is found along the left side of this chart. The second base is at the top of the chart. The third base in the codon is found along the right side of the chart. Let's do some examples. Here is a strand of RNA with the bases separated into codons. Given the first codon, AUG, We use the chart to find which amino acid would be coded for in the amino acid sequence. The first base is A, so it would be found somewhere in this row on the chart. The second letter is U, so it would be found somewhere in this column. This row and this column intersect in this square, so the correct amino acid is found somewhere in this square. The last base in the codon, G, narrows it down for us. The correct amino acid for this codon is methionine. This is the start codon and will initiate most proteins. Let's try another one. The second codon in this messenger RNA is ACG. The first letter is A, so the correct amino acid will be found somewhere in this row. The second letter is C, so it'll be found somewhere in this column. We can see that this row and this column intercept in this box here. You can see that there's only one amino acid in this box, so really the third letter, which is G, doesn't really matter because regardless of the third letter, the correct amino acid will be threonine, abbreviated as THR. The third codon is GAG. The first letter G, second letter A. The correct amino acid is somewhere in this box. We decide between those two amino acids by looking at the final letter G, so we know that the correct amino acid is glutamine, GLU. Let's skip to the last codon, codon 7, to finish up our examples here. Codon 7 reads UAG. First letter is U in this row. Second letter is A in this column. The third letter is G. This gives us a stop codon, which would signal the end of the gene and the end of translation. At this point, the newly formed polypeptide would be released. The universal genetic code can also be represented in a circular table that looks like this. Again, it may look complex, but is actually quite simple once you know how to use it. The first base is in the center of the circle. The second base is in the next circle, and the third base is in the outermost circle. Let's do some examples. Codon 4 reads CUU. The first base, C, lets us know that the correct amino acid is found in this quadrant of the chart. The second letter, U, narrows it down to this. You can see that the third letter doesn't really matter here. Regardless of whether it's a U, a C, an A, or a G, the correct amino acid will be leucine. Here's another example, codon 6, AGC. The first base, A, lets us know that the correct amino acid is found in this quadrant of the circle chart. The second base, G, narrows it down. The third base, C, lets us know that the correct amino acid is serine. Regardless of which chart you use, this genetic code is called universal because it is the same for all organisms. Whether you are a bacterium, a pineapple plant, a fish, or a human, each codon codes for the same specific amino acid. For example, the codon UUG codes for the amino acid leucine no matter what your species is. The universal genetic code represents strong evidence supporting the theory of evolution and the idea that all life shares one common ancestor. That common evolutionary ancestor must have had this genetic code and passed it on to all its descendants, the numerous species we see alive today. Yet another player in the process of translation is transfer RNA, or tRNA for short. Transfer RNA are single strands of RNA folded up into a compact shape. At one end, specific amino acids are attached to the tRNA. The tRNA brings the proper amino acids to the ribosome to be linked together in a chain called a polypeptide. A polypeptide is a long string of amino acids that will eventually fold to become a protein. In order to put the amino acids in the correct order, the tRNA has what is called an anticodon, a series of 3 bases that is complementary to the codon on the messenger RNA. The anticodon on the transfer RNA fits into the codon on the messenger RNA like a puzzle piece. Ensuring that the anticodon and the codon pair up correctly is how the proper sequence of amino acids is created. Let's see an example of how codon and anticodon fit together to ensure that the correct amino acid is added. Let's say that your first codon on the messenger RNA is AUG. A transfer RNA molecule with the anticodon UAC would fit perfectly and carry with it the amino acid methionine. If the next codon was GAG, a transfer RNA with the anticodon CUC would match and bring the amino acid glutamic acid. The methionine and glutamic acid would then bind together using a peptide bond, becoming the first link in the polypeptide chain. Transfer RNA would continue bringing amino acids to the growing polypeptide chain. Each amino acid would bond to the previous one using a peptide bond. Once the ribosome reaches a stop codon, the polypeptide is complete. It detaches from the RNA ribosome complex and becomes folded into a specific shape that will determine its function as a protein. The proteins produced by transcription and translation give us the traits we possess, from pigments that give us hair and eye color to enzymes that can determine dietary restrictions. In order to be expressed, these traits coded in DNA must be transcribed to messenger RNA and then translated to proteins. Now that you have an overview of the process involved in taking genes to proteins, let's review and add just a bit more information. We start with a strand of DNA in the nucleus. RNA polymerase binds to the promoter site on the DNA and begins transcription. A strand of messenger RNA is produced that is complementary to the nucleotide bases on the DNA. Messenger RNA leaves the nucleus, bringing with it the genetic message from the DNA. Once in the cytoplasm, the messenger RNA binds to a ribosome complex made up of two subunits, one large subunit and one small subunit. A transfer RNA carrying methionine docks to the start codon of the messenger RNA. The tRNA is docking at the peptidyl or protein site on the large ribosomal subunit. This site is called the P site for short. The anticodon of the tRNA is complementary to the codon of the mRNA to ensure that the proper amino acid has arrived. Next to the P site is the accepting or A site on the ribosome. The next transfer RNA will dock here to bring a second amino acid to the chain. Once the two transfer RNAs are in place, a peptide bond forms between the amino acids, and a new link in the polypeptide chain is created. The ribosome then moves down to read the next codon on the mRNA. This pushes the first transfer RNA from the P site to the exit or E site, and it is released. The second transfer RNA has moved to the P site, leaving the A site available to accept the next transfer RNA. The tRNA with the appropriate anticodon will dock to the codon in the A site, bringing with it the correct amino acid for the chain. A peptide bond forms between the adjacent amino acids, and the ribosome again moves down to read the next codon. The tRNA in the E site is released, and a new transfer RNA moves into the A site. The peptide bonds continue forming between adjacent amino acids as the polypeptide chain grows. The ribosome continues moving down the messenger RNA strand until a stop codon is reached. At that point, the ribosomal subunits fall apart and the protein is released. Another ribosome can find the start codon on the messenger RNA and begin making another protein from the same strand of messenger RNA. In fact, many ribosomes can bind and translate proteins from the same messenger RNA strand all at once. By doing this, your body can produce many copies of the same protein at a rapid pace. Generally, the processes of transcription and translation occur without error. Occasionally, however, a mistake is made. This is called a mutation. We will discuss mutations in the next lesson.